Hi friends, welcome back to my channel. In this video, we will discuss about magnetic nanoparticle-based nucleic acid isolation techniques, understanding principle and applications. If you are new to this channel, consider subscribing. So let's start with the video. Overview of the nucleic acid isolation. Nucleic acid isolation is a fundamental process in the molecular biology that involves extraction of DNA or RNA from the biological samples. And this procedure is very crucial for various applications like PCR, sequencing, cloning, and gene expression analysis. By isolating nucleic acid, we can study the genetic information, identify mutation, and understand the molecular mechanism underlying the biological process. So, nucleic acid isolation plays a key role or a pivotal role in molecular biology research. And nucleic acid isolation provide us with pure and intact nucleic acid DNA or RNA for analysis. The accurate isolation of nucleic acids is very essential for obtaining reliable results in the experiments like gene expression studies, genotyping, and genetic engineering studies. So it is very important to get pure and clean samples. The isolated nucleic acid will serve as a starting material for a wide range of downstream applications that contribute to advancement in the field of biotechnology, medicine, and genetics. So to study the mechanisms or underlying mutations, we need a pure nucleic acid that need to be isolated from a variety of sample types. So magnetic nanoparticle based technique have revolutionized the process of nucleic acid isolation by offering a rapid, efficient, and high throughput method. These techniques utilize the magnetic nanoparticles coated with a specific ligand that selectively bind to the nucleic acid and enabling the isolation from complex biological samples. By applying external magnetic field, we can easily separate the nucleic acid bound nanoparticle from other cellular components, simplifying the purification process. So incorporating magnetic nanoparticle based techniques into the nucleic acid workflow enhances the speed and yield of DNA or RNA extraction while minimizing contamination risk. So these innovative methods have significantly improved the efficiency and reliability of the nucleic acid isolation procedure in molecular biology research, and thereby providing pure and good yield of nucleic acids isolated from the complex samples. Now let's look at the traditional methods of DNA extraction and their limitations. So the traditional methods of extracting DNA is there from decades, and it has played a crucial role in various biological studies, and we can see two common approaches and their limitations. One is the phenol chloroform extraction, and this is a classic method that utilizes organic solvents like phenol and chloroform, and it breaks down cell membranes, releasing DNA. These solvents then separate DNA from proteins and other cellular components. The second method is the silica membrane based kits, and these are commercially available as kits which offer more user friendly approach, and they employ silica membrane that bind DNA while allowing impurities to pass through. This method is often faster than the phenol chloroform extraction. So examples for silica membrane based kit include can see from QIAGEN and other manufacturers. They provide silica membrane based kits. So what are the limitations of this traditional approach? One is the time consuming method, and it is labor intensive, and there is a risk of sample loss. Both the methods involve multiple steps like cell lysis, enzymatic treatment, washing procedures, and these can add up to significant time, especially when processing numerous samples. And these processes are labor intensive, meaning that the traditional methods require careful handling and precise measurement of throughout the process. We need to be you need to get involved into this. It cannot be automated, so this process cannot be automated. That is one disadvantage of this. So even though there are limitations, the traditional methods still have their place, and they can be particularly useful for getting high quality DNA. This gives very good yield of the DNA, which is high pure. Especially the silica column based methods is widely used in laboratories to yield high quality DNA. So now let's look at the molecular mechanism of magnetic nanoparticle based nucleic acid isolation. The molecular mechanism of MNP, or the magnetic nanoparticle based nucleic acid isolation, involves a combination of physical and chemical interaction between the magnetic nanoparticles, the target nucleic acids, and the solution environment. So let's look slightly deeper into this. We can explain this through surface recognition mechanism. So this involves the electrostatic interaction, covalent attachment, etc. So the electrostatic interaction, if you look at, this is the primary driving force for most of the MNP based nucleic acid isolation. The MNPs are typically functionalized with positively charged or polar groups on their surface like amine or carboxylic acid, and these groups interact with the negatively charged phosphate backbone of the nucleic acid through electrostatic interaction. The other approach is covalent attachment, for enhanced selectivity. The biorecognition molecules like antibodies or short DNA probes can be covalently attached to the MNP surface, and these molecules can specifically, bind and to the unique sequence present on the target nucleic acid, offering much stronger and more specific interaction compared to just the electrostatic interaction. So electrostatic interaction involves the selection of total nucleic acid. Here you can specifically identify the sequence of interest and capture that. So that is the difference between electrostatic interaction and the other method, which uses the covalent attachment of specific DNA probes to the nanoparticles. And depending on the type of surface functionalization and the strength of interaction, the binding of the nucleic acid to the MNP might induce some conformational changes in the nucleic acid. For example, the molecule might slightly bend or fold to optimize the interaction with the MNP surface. The other important factor that need to be considered is the solution conditions like pH and ionic strength. The acid-base properties of the functional groups of the MNP surface and the phosphate groups of nucleic acids are very crucial. Buffers are used to maintain the specific pH that favors the electrostatic interaction between them. And typically a slightly acidic pH around 6 to 7 is the optimal pH for binding to the magnetic nanoparticle. Ionic strength is another important consideration which we need to look into. The concentration of the salts in the solution that play a key role. High salt concentration can shield the electrostatic interaction between the MNP and the nucleic acid, reducing the binding efficiency. And therefore, buffers with controlled ionic strength are required for optimal binding. So this need to be experimentally determined to see how well the nanoparticles and the nucleic acid interact. So we can check the flow through after the binding condition. Flow through of that can be checked to see the presence of loss of DNA in the unbound, to understand how much binding is happening in this condition, solution condition. So ionic strength and pH can be varied to get the optimal binding. Once the binding between MNP and the nucleic acid occurs, an external magnetic field is applied, and these magnetic properties of the MNP can cause them to migrate towards the magnet and pulling the attached nucleic acid along with it. So the separation relies on difference in the magnetic properties between the MNPs and the rest of the sample components. So typically the magnetic nanoparticles can be magnetized using a magnet, and other components will get separated because the nucleic acid will be bound to the nanoparticle, and the rest of the cellular components can be thrown out. So the unbound cellular debris, proteins and other sample components remain in the supermagnet, and which allow for easy separation. So depending on the application, the purified nucleic acid might need to be released from the MNPs. So this can be achieved through various methods. One of the method is changing the pH, as mentioned. So pH plays a key role in holding on the magnetic nanoparticle holding on to the nucleic acids. So by adjusting the pH to a different range can interrupt the electrostatic interaction between MNPs and the nucleic acid, and which can cause them to dissociate. Elution buffers like containing chelating agents can be used to disrupt binding between functional groups and the nucleic acid. For example, TRICDTA buffers can be used at a pH of 8 to 8.5 to release the bound nucleic acid from the MNPs. So other approaches include enzymatic cleavage. If the biorecognition of the molecules were covalently attached, specific enzyme that can cleave the bond between the molecule and the nucleic acid can be used for elution. So these are applicable when we use a covalent based attachment strategy. The molecular mechanism relies combination of tailored surface chemistry, manipulation of solution condition, and the unique magnetic properties of the MNPs to achieve the selective and efficient isolation of the target nucleic acid from complex biological samples. So biological samples can be blood, plasma, serum, stool samples, and other cellular tissues, and variety of samples can be used to get a nucleic acid isolated from these samples. So each of these requires a specific lysis step. Some of the bacteria or other difficult to lyse samples requires a high lysis condition or heating or even sonication to disrupt the cellular components and release the nucleic acid into the solution. So let's look at the workflow of magnetic nanoparticle based isolation. The magnetic nanoparticle based isolation offers a very fast and efficient way to isolate the nucleic acid from various samples. As I mentioned, there are a variety of sample types which we can use to isolate the nucleic acid. First step in the workflow is the sample preparation. This is the initial step that involves the preparing of the sample to release target nucleic acid. There are a specific method depending on the sample types. For example, if you are using cells, this is often cell lysis that is involved, which breaks down the cell membrane and releases the cellular contents including nucleic acid. and techniques like mechanical disruption, enzymatic lysis, or detergents can be used. So this depends on what type of initial sample you are using for the isolation of nucleic acid. For tissues, homogenization might be required to break down the tissue into fine suspension. And for biological fluids like blood or plasma might require centrifugation to remove the debris, or treatment with protease K or proteinase K may be required as a pre-processing step before the lysis step, or along with the lysis we may have to use proteinase K in the workflow. So once the sample is prepared, next step is the binding of nucleic acid to the MNP. So MNPs functionalized for capturing the nucleic acid are added to the prepared sample, and solution conditions like pH and ionic strength are carefully controlled to optimize the interaction between negatively charged nucleic acid and the functional group on the MNP surface. This typically involves using buffers with high pH and controlled salt concentration. And depending on the type of MNP, the binding can occur through electrostatic interaction or covalent attachment. So we have discussed this previously. So it can be electrostatic interaction or the covalent attachment. So once it is bound, once the nucleic acid is bound to the MNPs, this need to be separated from the other components. So after allowing sufficient time for binding to occur, an external magnet is applied, and this MNP now attached to the target nucleic acid migrate towards the magnet due to their magnetic properties, and the unbound cellular debris and proteins and other impurities remain in the supertape. And this separation is very rapid and efficient due to strong magnetic attraction. People often use ethanol also to help in the binding of the nanoparticle to the nucleic acid. So along with the buffer condition, we have to consider the use of alcohol for the efficient binding of the nanoparticle and the nucleic acid. So once the magnetic nanoparticles are separated out from the bulk of the solution, washing need to be done to remove the impurities. The isolated MNP nucleic acid complexes are washed with buffers specifically formulated to remove remaining impurities like cellular debris, proteins and salt. So a lot of lipids and proteins and other components from the sample can interfere, so that need to be washed out. The number of washing step might vary, and that depend on the specific protocols and the desired level of purity. So this need to be tweaked to an optimal level so that you get a cleaner nucleic acid after isolation. So after washing, we need to elute the bound nanoparticles. Depending on the downstream application, the purified nucleic acid might need to be released from the MNPs. And this is achieved through a variety of methods by changing the pH or elution buffer, changing the concentration of the buffer, enzymatic cleavage. So all these we have discussed. So depending on the specific use case, we have to use the method suitable for that purpose, and which can elute the DNA out from the magnetic particle. So following elution, the purified nucleic acids are ready for further analysis or downstream application like PCR, sequencing or gene expression studies. So you can check the purity of the isolated nucleic acids using nanodrop or other methods. You can run even a gel to see the purity, and we can check the nanodrop reading to understand the 260 by 280 ratio and identify how pure it is, and also to look at different shoulder peaks, whether it has of the impurities which is present. So let's look at the advantages of using magnetic nanoparticle based nucleic acid isolation. The magnetic nanoparticle based nucleic acid isolation offers several advantages over the traditional methods. So we have seen in the beginning that what are the traditional methods which we use for nucleic acid isolation, but magnetic nanoparticle offers some advantages over the traditional methods. One is it is very rapid and it requires reduced time compared to traditional methods like phenolchloroform extraction or centrifugation. MNP isolation is significantly faster, and the magnetic separation step allow for rapid isolation of the target molecules, significantly reducing the processing time. This is particularly beneficial for high throughput application where processing numerous samples are necessary. So this is one advantage, is that it is rapid and it supports high throughput sample processing. Second is high yield. The traditional methods often involve multiple steps with the potential for sample loss during the transfer and manipulation, whereas MNP isolation offers a streamlined process with minimal handling steps. The magnetic separation ensures efficient capture of the target molecules, minimizing the sample loss and maximizing the yield. The other advantage is the automation compatible. The magnetic separation step readily lends itself to automation, so it can be automated. So no manual processing is required. User can just add in the sample to the desired well, and it can be automated by a robotic system. The automation capability makes the MNP isolation ideal for processing large number of samples efficiently and consistently, and a crucial factor in the high throughput application like clinical diagnostics. So where you have a large number of samples which you need to process, it can be automated, and it can be done very quickly with high throughput. The magnetic nanoparticle based extraction is very versatile, and it is compatible with various sample types like blood, cells, tissue, etc. The isolation protocols can be adapted to work with a wide range of sample types, and by adjusting the sample preparation step and optimizing the MNP surface chemistry, one can isolate nucleic acid from blood, tissue, cultured cells, and a number of sample types. We can use this method for isolating nucleic acid from different sample types, and this versatility makes the MNP isolation a valuable tool for various research areas. The magnetic nanoparticle isolation can yield highly purified nucleic acid with minimal contamination. This is another advantage, that the purity of the isolated nucleic acids are much higher compared to other methods, and it is very much scalable. The technique can be easily scaled up or down to handle different sample volumes, and it is very safe compared to other methods like using harsh organic solvents. MNP isolation offers a safer alternative. So now we have seen a comparison between the traditional method, advantages, principle. Let's look at the applications of nanoparticle based isolation. So the magnetic nanoparticle based isolation has revolutionized the field of nucleic acid isolation due to its speed, efficiency, and versatility. Coming to genomic studies, MNP isolation is a valuable tool for researchers studying the entire genomes because of its rapid and efficient isolation of high quality DNA from various sources. The purified DNA can be used for downstream applications like sequencing, gene mapping, and other comparative genomics. It is also widely used to study mRNA and in transcriptomics, and it is widely used in the epigenetic studies and also in the clinical diagnostics. The pathogen detection and the cancer biomarker analysis, MNP plays a crucial role for isolating the high pure DNA from these different sample types. It is also used in forensics for DNA profiling. So these are some of the applications wherein we can implement MNP based isolation techniques. So coming to the comparison, if you can see this table, it is much faster compared to the traditional method. MNP based nanoparticle extraction method is much faster compared to the traditional methods. The yield and the efficiency is much higher in magnetic nanoparticle based extraction, and it is often highly compatible with automation, whereas traditional methods are less compatible. The cost P method is cost effective for the high throughput application, whereas traditional methods only cost effective for low throughput application. Purity of the nucleic acid isolated through MNP method is high pure, whereas the traditional methods might require additional purification step. Overall method is much safer because it is not using very harmful chemicals, whereas the traditional methods like phenylchloroform can involve hazardous chemicals. So these are the overview of comparison between the MNP based isolation and the traditional methods. Next, coming to the challenges of MNP based nucleic acid isolation technique. So what are the challenges? So even though it offers very numerous advantages, there are some challenges also along with the magnetic nanoparticle based systems. One of the issue is the nanoparticle stability. So there are two things which we need to consider: the aggregation and the storage. So aggregation means MNPs can clump together if not properly stabilized, and this can hinder their ability to interact with the nucleic acid and affect the separation efficiency. So this is one of the problem, that the clumping effect or the aggregation of the nanoparticle can impact on the nucleic acid binding efficiency, and MNPs can degrade or lose their magnetic properties over time. So proper storage condition and choosing MNPs with good stability is very crucial for consistent performance. So these two are some of the challenges, the stability. Coming to the binding and elution condition, specificity, the finding the right balance between specific binding of the target nucleic acid and the minimizing non-specific interaction with other cellular components is also very important, because there is a crucial overlap between the pH and ionic strength, wherein the nucleic acid binds and other components does not bind. We need to find a balance between these two. So sometimes it is tricky because of the complex nature of the biological samples. We may need to titrate some of these components to get an optimal binding and efficient purification. So elution efficiency, releasing the purified nucleic acid from the MNP without compromising their integrity can be a little challenging. Different elution methods like changing the pH, specific buffers, enzymatic cleavage might be required depending on type of the MNP and the downstream application used. So These are some of the challenges. In certain cases, additional purification step may be required, like sequencing and other downstream processing might require additional purification step even from the MNP based purification system. So if you look at the cost effectiveness, initial investment MNP isolation kits or reagents might have a higher upfront cost compared to the traditional methods. So if you're planning to automate, we need to buy a robotic system and other things. So initial investment will be much higher compared to the traditional methods, but for the high throughput it will be really useful. It will be really cost effective. So how to overcome these challenges? One of the strategy is to choose high quality MNP with proper surface coating and storage recommendations. So this can overcome the problem with the clumping and storage, and carefully optimizing the binding and elution conditions based on the target nucleic acid and the downstream application. So you know that already that okay, you're going to use this application for purified nucleic acid for certain applications. So you can optimize this condition. Utilizing commercially available MNP kits are often come pre-optimized for specific sample types, so which is much easier to work with. Getting a commercial kit which would have already workflow optimized for that particular sample types, and cost benefit analysis to determine the advantage of MNP isolation outweigh the initial investment, because for the high throughput sample it is really useful. We can consider the cost benefit analysis and see how well it will fit into their budgeting, and it will be cost effective compared to the traditional methods. What are the recent advances in the magnetic nanoparticle based nucleic acid isolation? So the magnetic nanoparticle based isolation is still continuing to evolve and offer exciting advancements that address some of the existing challenges and open doors for new possibilities. Some of the examples include enhanced functionalization strategies. For example, targeting specificity. We can develop novel surface functional strategies for MNPs. This includes attaching antibodies or aptamers with high affinity for specific nucleic acid sequences. These targeted MNPs can significantly improve the selectivity of isolation, minimizing the non-specific binding of unwanted molecules. So certain examples like isolation of cell-free DNA. If you want to target certain pieces of information from the complex sample matrix, we can use specified oligoprobes attached to the MNP, which can capture the low abundant sequence and concentrate it instead of capturing the total DNA. Multifunctional coatings, and the MNPs are being designed with multifunctional coating, and these coatings can combine functionalities like targeting molecules, spacer molecule to optimize the binding distance and responsive moieties for controlled release of the captured nucleic acid, and this allow for tailored MNPs with improved performance for the specific application. So we can tweak these conditions for specific applications. As mentioned earlier, it is really useful for integrating with the microfluidics and lab on chip devices. The MNP isolation is being integrated with microfluidic devices and lab on chip platforms. So for point of care applications, it is really useful. This workflow is really adaptable for integrated chip processing and also for utilizing in the point of care applications. Nowadays, new MNP compositions are being explored using the biocompatible materials like iron oxide or even the non-metallic nanoparticles, and this focus on the biocompatibility ensures the safety of MNPs for potential in vivo application and downstream biological studies. Researchers are developing MNPs with the improved magnetic properties for enhanced separation efficiency, and this allows for faster and more complete isolation of the target nucleic acid, minimizing the processing times and potential sample loss. So we have seen different methods and the principles and the how this nucleic acid is isolated. What are the future perspectives for magnetic nanoparticle based nucleic acid isolation? So the MNP based isolation offers a powerful and versatile approach for nucleic acid isolation, and as research continues, we can expect more advancements that can broaden its application and address existing challenges. So one of the area everybody is keen on is the point of care diagnostics, because MNP isolation its rapid, efficient nature makes it suitable for developing point of care diagnostic test, and which is even adaptable for the microfluidic based approach. So miniaturized and portable devices utilizing the magnetic nanoparticle could enable rapid detection of infectious diseases, genetic mutation, and other toxin detection at the point of care, which can improve the early diagnosis and the treatment interventions. So it is also useful for environmental monitoring by targeting specific DNA or RNA sequence from microorganisms in the environmental samples. Research can access the microbial diversity, detect pathogens, and monitor environmental health. Some of the emerging technologies like CRISPR, the revolutionary CRISPR gene editing technology can potentially be combined with the MNP isolation for targeted manipulation of genes, and the MNPs could be functionalized to deliver CRISPR components to the specific cells, allowing for precise control over gene editing process. Nanotechnology offers exciting possibilities for further development of MNPs, and the novel nanomaterials with unique properties can be used to create MNPs with enhanced functionalities for improved targeting capabilities. So while there are challenges, we can even standardize and reduce the cost of the MNPs and to make them user friendly, so user can use it much cheaper and cost effective way and in a rapid fashion to get efficient nucleic acid isolation. So overall, the magnetic nanoparticle based isolation has emerged as a powerful technique for isolating the nucleic acids, both DNA RNA, from variety of biological samples, and this approach offers a more faster, more efficient and versatile alternative to the traditional methods. So MNPs functionalized with specific capture molecules interact with the negatively charged phosphate backbone of the nucleic acid, and an external magnetic field allows for rapid separation of MNP nucleic acid complexes from the rest of the sample, and the elution methods like changing pH or specific buffers can release the purified nucleic acid from the MNPs. So let's look at the potential impact on research and diagnostics. The MNP isolation streamlines the research workflow, saving time and minimizing the sample loss, and this allows researchers to delve deeper into the various fields of molecular biology. And the high purity and efficiency of MNP isolation is very crucial for advanced technique like next generation sequencing and gene editing using CRISPR technology. So in the area of diagnostics, MNP isolation holds a promise for developing rapid point of care tests for detecting infectious diseases, genetic abnormalities, and environmental toxins. So continued improvements and innovation and optimization of the process and the workflow can further enhance the user friendliness and cost effectiveness of this technique, whereby we can have a very high quality nucleic acid isolated with improved efficiency, faster methods, and very versatile, and that can be suitable for point of care application with advanced automation condition for handling much larger sample types. So these are some of the advantages and overview of the MNP based nucleic acid isolation techniques. So you can refer these links and the papers for further reading. I will share some of these in the description as well. You can go through this, and if you like the video, please press the like button and share it with your friends. Thank you.